Hi, and welcome to Meet the Microbiologist, the podcast about the people who discover, innovate, and advance the field of microbiology. I'm your host, Ashley Hagen, and today I'm joined by Dr. Neil Mabbott, professor and personal chair of immunopathology at the University of Edinburgh. Welcome to the show, Neil.
It's good to speak to you.
We're so happy to have you. You study the pathogenesis of infectious diseases within the immune system, with a particular interest in understanding host pathogen interactions within the mucosal immune system, and a focus on prion diseases and other gastrointestinal pathogens. So let's start there. Can you give us a description? What are prions?
That's a really good question, and it's something that's been occupying our minds for decades, in fact. I mean, there have been several Nobel Prizes awarded in this field, and still amongst the field, we still argue about what a prion actually is. But I guess we're slowly reaching consensus in this, and I say slowly because there'll always be somebody who disagrees with us, and science is ever correcting, etc. But we really consider now prions to be infectious proteins. So prion, every cell in our body expresses the normal cellular form of the prion protein, and we really don't really know what this protein does. It's highly conserved in mammals, expressed on most cells in our body. Some cells express really highly, so cells such as neurons. During prion disease, this protein starts to abnormally fold. So normally the protein is expressed in a predominantly alpha helical form, so it has those kind of curly tubes. But during prion disease, it starts to misfold and becomes more beta-pleated sheet. And when that happens, it confers some really interesting biochemical and biological properties to that protein. Whereas the normal cellular form of the protein is expressed on the cell membrane, it's soluble in detergents, it's relatively easily digested by proteinases, etc. The abnormally folded form, which we find during prion disease, now forms aggregates, often in the extracellular environment. It's also becomes very resistant now to standard decontamination procedures, relatively resistant to proteinases, but most importantly, it becomes infectious. And by that, I mean that if we were to inject some of those proteins into a recipient animal or indeed person, those proteins can, by some unknown mechanism, or I say unknown, but we are obviously studying that, but the precise mechanism is not known. By some unknown mechanism, can convert the normal cellular form into the infectious form. So you get amplification of those prions, those prion proteins, and they can transmit disease. And ultimately, if left unchecked, can lead to severe damage of the neurons in the brain of those animals or people.
So you mentioned that these proteins are infectious if they are injected, maybe into a new host. Can they be transmitted naturally outside of injection from host to host as well?
Yeah, so I described the experimental situation, but of course, as microbiologists and scientists, we're interested in natural disease. And that's the area that I've been studying for at least 2 decades now, is how do the prions go from the site of infection to the brain where they cause disease? And when we think about prion diseases, a predominant natural route of transmission is the oral route. So by ingesting or eating infectious prions, and those can be sheep, for example, on pasture in fields, maybe eating some contaminated pasture, etc., which is contaminated with prions. They enter the digestive tract. Prion disease is a neurological disease in the CNS. It can cause extensive damage to neurons in certain regions of the brain. And we've been really interested in how do those prions go from the gut or the intestine to the brain, where they eventually cause disease. And this became really, really relevant when I started working on prion diseases. I started working in the late 1990s. And up until then, for the majority of the time, I guess prion disease had been perhaps a biological curiosity. The predominant disease of interest at that time was scrapie in sheep.
Mm.
Um, and at a similar time, actually, people were interested in an interesting disease known as kuru, which was identified by or recognized by scientists in uh, an indigenous tribe in Papua New Guinea. It was a neurological disease which was spreading amongst members of this group of individuals. When scientists went to study this disease, they made a link between transmission of disease and cannibalistic rituals. And what they were observing was members of the tribe, when somebody died, there was a kind of a cannibalistic ritual. They ate their elders. I guess it was just a sign of respect, etc. But what they noticed was only certain individuals were fed the brains or the nervous tissues from these individuals, and that tended to be the women and the young people, whereas the men, they ate the muscles, etc., etc. They made the link between the contamination of the brains from these individuals and transmission of disease, and that was really, really important to put a demonstration that prion diseases could be transmissible, and indeed, a Nobel Prize was awarded to the person who made that link. So those were those two diseases. But I guess during the 1980s, BSE was identified as bovine spongiform encephalopathy, so a novel prion disease in cattle, and it was spreading like wildfire in UK cattle, spreading. Many tens of thousands of cattle succumbed to bovine spongiform encephalopathy. And it was considered most likely this occurred due to feeding practices at the time through use of meat and bone meal. And that was derived from, or could be derived from, materials from sheep, etc. And it's possible that material from an infected sheep entered into this meat and bone meal material, which may have had a BSE-like prion in it, and that then was most likely propagated through the BSE herd through consumption of this meat and bone meal. But at the time, there was concern that this novel prion disease might also transmit to humans through the consumption of BSE-affected meat. And unfortunately, I think it was 1996, that appeared to be the case. And I remember this very well. It was very striking because I'd literally just started working on prion disease as a young postdoc at the time. And it was announced in the Houses of Parliament in the UK, I think in the early summer of 1996, the scientists in Edinburgh identified a novel prion disease in humans. At the time, it was known as new variant CGD. We now call it variant Creutzfeldt-Jakob disease. And the indications were that this was most likely due to consumption of BSE. And this disease was striking because it was affecting young people. This was affecting teenagers, people in the early 20s, etc. And for a human prion disease, this was very rare. At the time, sporadic CJD was recognized in people, but this tends to affect elderly people or people at least in their 60s. Very rare to see this in young people. So it's very striking that this prion disease was identified in young people, certainly teenagers or those in their 20s. And later, when people came to study the pathology of this disease, it had a unique pathology. But then when further studies were shown, it was shown to be very, very similar to a BSE-like disease, suggesting that our concerns were real and that BSE perhaps had been transmitted to humans through the consumption of infected materials. So, as you can imagine, interest in how prions are spread became very prominent, and there was lots and lots of interest in this area of research. It became a very exciting period to be working on prion diseases.
Yeah, understandably. And what is some of the pathology that was observed in those two diseases and where there were similarities?
Okay, yeah. So, I mean, this is one of the really striking things about prion disease. So, I discussed to you earlier how prions are infectious proteins,
Mm-hmm.
or at least we think that they are predominantly infectious proteins. And what was really interesting was that if we took BSE prions, so these are prions from cows,
Yep.
and injected them into laboratory mice, we see a specific set of pathological signs in the brains of those animals. The main one being spongiform pathology. So by that we mean the brain essentially looks like a sponge.
Mm-hmm.
So we see lots of holes in what we call the neuropil of the brain. I'm sure many will have seen those classical kind of neuropathological images where the
Mm-hmm.
brain essentially looks like a sponge. Now this targeting is not random. The prions target very specific regions of the brain in those recipient animals, and it's very reproducible. That helps us because we can then type the particular strain of prion. We can say, okay, this has a particular pattern of damage in the brain, so that looks like BSE prions. Or this has a different style of targeting that looks like chronic wasting disease prions, etc., etc., etc. Now, what was very interesting was when uh, researchers in Edinburgh took prions from cows, injected them into mice, you get a particular type of neuropathology. When they also took those prions from these new cases of CJD and injected those into the mice. So these are human prions which have been injected into mice. They looked identical to BSE
Wow.
in the brains of those mice. So what happened here is BSE prions had gone into humans. So those cow proteins had converted the human prion protein into a new human prion. Those
Wow.
human prions are then been injected into mice, and those human prions have converted the mouse prion protein into prions. But throughout that process, they still look like BSE.
Interesting.
And so I still find that really remarkable. This protein, it doesn't appear to have doesn't have any DNA or RNA there to convey all this kind of information, is able to convey these the similarity throughout this chain of events, and it's highly reproducible. They
Wow.
don't diverge, etc. If you keep doing these experiments, you get very precise neuropathological targeting in the brain. And
Wow.
likewise, as I mentioned, you see specific patterns of damage in the brain, and you also see specific patterns of this buildup of these abnormal prions. They accumulate in very specific patterns depending on the disease you're working with.
Wow. That is really interesting. So when you entered the field, as you mentioned, this was a really sort of exploding topic. What were some of the key questions that were on the table?
Well, the key questions, of course, at that time were how bad is this going to be?
Mm-hmm.
And should we be concerned?
Mm-hmm.
Obviously, it was very concerning, of course, and very worrying that at the time just a handful of cases had been identified. And what we didn't know was, were these going to be the only ones? Are these the only unfortunate individuals that are going to be infected? Could it be tens? Could it be hundreds? Could it be
Mm-hmm.
thousands? Or could it be millions? And we really didn't know that. So the government and all the administrations, etc., had some real key questions for this. How bad was this going to be? And of course, how can we diagnose it? How can we test if somebody has the disease?
Mm-hmm.
Can we treat it?
Mm-hmm.
And of course, lots of other risk factors as well, which tissues would likely to be infected, etc., etc. And these are
Mm-hmm.
still the same kind of questions that we're asking to these days as well. So they're very difficult diseases to work in. We can't look down a microscope and count prions.
Mm-hmm.
They are individual proteins or groups of proteins. It's not like working with a salmonella or a virus, etc. Also,
Mm-hmm.
we can't use tools like PCR or genomics to study these agents because they don't have a genome.
Right.
So it makes working
Right.
with these things very, very difficult. And of course, as well as these kind of public health issues, as well as food security issues as well, many were also interested in how can we stop the disease spreading in cattle, etc. And there were some
Mhm.
very key experiments or suggestions from colleagues like Charles Wildsmith, etc., who were instrumental in identifying the link between meat and bone meal, etc. And so that practice was stopped. We
Mm.
also removed particular tissues from the food chain as well. Those tissues, so research was done to identify which tissues contain the most amount of prions in a BSE infected animal. They were removed from the human food chain as well, all to reduce the likelihood of onward transmission of disease, etc.
Mhm. So you
Then, of
mentioned with
course,
the
there was the concern. Okay,
oh,
sorry,
sorry,
yes, please.
go ahead. I don't,
Yeah,
this is
so
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 fascinating. So I don't want to cut you off. Please keep going. I'll remember
no, no,
my
no. So
questions.
we were obviously
Yeah.
very interested in BSE in cattle, but of course, and BSE had spread to humans. Well, what are the chances it could spread to other animals as well? And of course,
Mhm.
cows aren't farmed in isolation. They tend not to be farmed in isolation on farms. They might be on pastures with cows or other animals. And so lots of studies were done to investigate the potential that BSE could transmit to other species as well. So many of my colleagues in Edinburgh and around the world have been studying whether BSE has a potential to spread to other animals, and if it does, perhaps through selective breeding, etc., or genetics, can we prevent or reduce the likelihood that disease might transmit to other animals? And it's interesting you say this is fascinating, etc. And that's always been the experience I've had working on these diseases. And it's been really interesting because when I started with my postdoc, these events hadn't happened, or the sorry, BSC was a key event, but it
Mm.
hadn't been identified that it transmitted to humans. And so there
Mm.
was interest, but nowhere
Yeah.
near as much as the interest became a couple of years later when I started working on that. So
Hmm.
actually, some of my friends, when I said I was moving to Edinburgh now, having done my PhD and I was going to be working on prion disease, they say, "You're working on prions? Why?
Mm.
What are prions, etc." Okay, well, good luck with that, Neil,
Right. Yeah.
etc. And then within a year or two, everybody was really, really interested. Oh, Neil, you're working on prions, and everything had something to
Mm.
say and suggestions for how I should be doing research, etc. And I remember it was really interesting. You'd meet somebody in a bar
Mm.
in the evening after you'd been in the lab, etc. And somebody would say, "Oh, what do you work on?" "Oh, I'm a postdoc now. I'm doing some research on a disease on prion diseases." And they say, "Oh, really interesting." And then we'd start chatting about the soccer or something else, etc. Yeah.
Mm.
And then once variant CGD was announced, etc. You mentioned work on prions. Everybody had something to tell you what you should be doing, etc.
Yes.
And I remember actually being in North America, being in an ice hockey game, and just chatting to somebody next to me in the audience during one of the breaks. It turned out
Mm.
this guy was a farmer, and he had lots to tell me about prion diseases and BSE, etc.
Wow.
So
Wow.
it's been useful to be able to work in a field where it feels you're doing something relevant.
I love that. That's so neat. And to sort of be evolving your work as the research is evolving, sort of cutting edge on a topic that is of so much public health importance. So I want to go back a
But
little
but
bit.
it's also a case. Sorry, so it's also a case of taking making use of the opportunities that come your way. Because as I pointed
Mm.
out, I chose it through scientific interest, not through public health
Yeah.
interest at the time.
Right.
But it became that, and I was able to kind of exploit that opportunity to
Hmm.
uh, throughout my research because it opened up many opportunities, and that came had it advantages when you were looking for collaborators on particular studies.
Hmm.
And I've
Yeah.
been fortunate to have many really interesting and helpful and supportive collaborators throughout my research. I couldn't have done many of my studies without collaborators. I say, "Oh, I'm working on this Crohn disease. I've read your study. It'd be great if we could
Mm.
collaborate and I could perhaps use your mouse model or use some of your reagents." People get back, "Oh, well, that's a really interesting project. I'd like to learn more about it." "Yeah, yeah, sure, we can
Yeah.
collaborate, etc., etc." So it's been very, very useful in many ways.
That's really neat. And a very important point, I think, to add on there, that it didn't start as a public health interest, but gathering public attention often requires that human disease, the transmission to humans. I feel like that is when it captures sort of the public attention. But for scientists, this background research and sort of laying the groundwork, COVID, I think, is another example of that. There was so much basic research into different coronaviruses and RNA viruses going on before SARS-CoV-2. And it's such a important point. I think that work helps us to be ready and positioned to respond when that need arises.
Exactly. And also as scientists, though, we also have a duty to help communicate what we're doing and why to folks as well. So
absolutely,
we can be involved in public engagement in all sorts of kinds of ways. Like I mentioned, just chatting to that guy in the bar.
Yeah.
When you're buying your newspaper and milk from the grocers in the morning, etc. Just explaining what you're doing and giving the relevance. And I always say it's really important because at the end of the day, taxpayers pay my wages through the grants I secure, etc.
Right.
So they have a right to learn how I'm spending their money and how it's being used.
Yeah, that is really important. The educational piece and just kind of the I think the relational aspect of that. So I want to go back a little bit, and you mentioned that when you're looking at the brain and the pathology of the brain, that you can see a buildup of these prion proteins, but also mentioned that it's difficult to count them and to view them sort of under a microscope. So I'd love to talk about how you do this. How do you visualize a protein? How do you study it in the lab? Some of the techniques that you use.
Yeah, so it depends on the kind of studies you're doing, but in our studies, we do lots of histopathology. So we collect tissues, and oftentimes we might use immunohistochemistry to detect them. So we do have antibodies against the prion protein.
Okay.
Unfortunately, because the prion protein is host encoded, we make the prion protein ourselves. It's very difficult, if not almost impossible, to make an antibody which is specific only to the infectious prion, because our immune systems create tolerance to the prion protein. However, that said, we can stain a tissue section with an antibody against the prion protein, and we can see those abnormal accumulations start to appear in those tissues. So by using immunohistochemistry, we can see the sites of prion accumulation. We can see those become more evident as the disease progresses.
Okay.
Now that might give an indication of sites of abnormal protein accumulation, but that might not necessarily always be infectious prions. So there are other techniques we need to do to discriminate, are these infectious prions or not?
Okay.
And oftentimes, if we really want to say, is there infectious prion here, we'd actually make an homogenate or a preparation from that tissue and inject it into recipient mice.
Okay.
And then study the time it takes for those mice to develop a prion disease. And that way we can then be certain there were infectious prions there, because we could take some material from one animal, inject it into a recipient animal, and show that there was transmission of the disease, and therefore there must have been infectious prions there. So that's two ways we can use. We can either use immunohistochemistry. Some would say the gold standard is, we call this the mouse biopsy, when you take material from one mouse, inject it into the others. Another way, we can use a Western blot as well. So we can make an homogenate of a tissue, run it through a gel, apply that to a nitrocellulose membrane, treat it with proteinase to destroy the cellular form of the prion protein, and hopefully what we're left with then, if there are prions there, is the abnormally folded version of the prion protein, which we then detect with an antibody. There is I guess I would say a more recent, although it's probably been out for about 10 or 15 years now, is essentially kind of a PCR for proteins. It's known as the PMCA assay, whereby it's an in vitro or a test tube test where you have your test homogenate. So you have a homogenate where you think you have prions in there, and you have essentially a suspension of normal prion protein. And what you do is you add your test proteins to the normal prion protein. You sonicate them, so you give them essentially a good shake. And what you're hoping is that if there are prions there, those prions will start to convert the normal cellular form into abnormal prions.
Hmm.
And you do multiple rounds of this kind of shaking. You break up the aggregates if there are any aggregates using the sonication, a bit more shaking. And with time, you can go from a very, very small amount of prions, which would be in your test sample, to a large amount that you're then able to detect on something like a Western blot. So we have multiple tools that we can now use to do that, to try and detect whether the prions are there or not.
Okay.
But it can be very difficult to determine whether you have BSE prions there, scrapie prions, prions, or a novel prion. So we often have to overlay that with different types of assay to be sure that this is a different type of prion or the type of prion that we're interested in.
So is the primary distinguishing factor between these different prion diseases the pathology, the way that it impacts the host, or is there a difference in folding that occurs in the protein itself?
There's never, if only we knew that.
Okay.
Unfortunately, there's never one fast rule. There's never one fast rule. There are
Mm-hmm.
multiple different ways in which so yes, there are multiple ways you can characterize a strain. One
Okay.
being the type of pathology it causes in the brain, so the
Mm-hmm.
amount of holes, etc. It could be determined by the deposition of that prime protein within the brain, or indeed within the body as well. So
Okay.
if we take, for example, sporadic CJD in humans, if we look at autopsy in a patient which had sporadic CJD, predominantly the prions would be accumulated in the CNS tissues of that individual. It would be very unlikely to detect significant buildup of prions in any other tissues in the body of that individual, even though they succumb to clinical disease. Contrast that though with somebody who had variant CJD, we see quite different accumulation. We do see the accumulation in the nervous tissue of that person. Also, we see accumulation of prions in the lymphoid tissues in the immune systems of those patients.
Hmm.
And that's important because the prions actually hijack, or we think the prions actually hijack the immune system to infect the disease. So the tissue targeting of the prions can vary quite distinctly depending on the disease. And that appears to be similar also between BSE in cattle and scrapie in sheep.
Hmm.
In that in
Interesting.
BSE in cattle, the disease is predominantly targeted to the nervous tissue, whereas in scrapie in sheep, you find the prions in many tissues throughout the body, depending on the time of the disease. Also, the duration of the disease can vary between strains. Some diseases will be very long, others reasonably short, and that can be the time from exposure of prions to developing disease, and also the clinical phase of the disease as well. Sometimes it can be very short, others reasonably long.
Hmm. So what
And
are the
also the
symptoms?
symptoms.
Yep.
The symptoms and signs can be
Mhm.
different as well.
Mhm. That is exactly where I was headed. So what are the symptoms of these diseases? You mentioned they may differ based on the type of disease, but can you give us some examples?
So the diseases are essentially associated with neurological decline.
Mm.
And in the very early stages, though, unfortunately, they can be quite non-specific. So if we take, for example, variant CGD, I mentioned it can affect young people. And in the very, very early stages, that could come across as feeling a little bit grumpy or down.
Mm.
And of course, we all feel grumpy or down at some point, don't we? And teenagers can feel grumpy or down most days, as far as I'm
Right.
aware as well.
Right.
So
Yeah. Mm.
disentangling that from just having an off day to actually this
Mhm.
is the very early signs of a prion disease can be quite difficult. And that can be true of many diseases. If we think, for example, between COVID and many other diseases, early disease would be flu symptoms. So is this flu? Is this COVID? Is it another disease? It can be very difficult to distinguish between that. But that said, if we go back to variant CGD, of course, it can rapidly progress to quite serious neurological decline to the stage where the patient can no longer feed itself, etc., and becomes completely hospitalized, relying on uh, uh, exogenous feeding, etc. You can get a very rapid, progressive neurological decline in those individuals. You'll have seen the images of bovine BSE infected cattle where they can't walk, etc., because you get very rapid loss of gait or ability to walk in those individuals.
So, of course, the next sort of natural question that's coming to my mind when I'm thinking about these early stages of identification of the disease, before we knew that it could be transmitted from cattle to humans, and before we knew that bone meal might be a potential source of transmission, were there any signs in the cattle that they were unwell? I'm thinking, at what point can this disease be transmitted? Is it in the early stages before those symptoms are really identifiable? Can it still be transmitted if, the bone meal is consumed by its counterparts, for example?
Okay, I see where you're coming from. Yeah, so BSE, so variant CGA, for example, in humans, we think probably had a 5-year duration from the time of ingestion to clinical disease. It's very difficult to pin it down exactly in a natural disease in something like human. We don't know the exact onset, but we think probably that type of duration. And for the majority of that period, the individual will look perfectly healthy. That said, for much of that time, the lymphoid tissues of that individual may have had high levels of prions within them. And it's not until you get significant, or tends not to be until you get significant accumulation of prions in the central nervous tissues that the individuals start to show clinical signs. And the same is true in an infected animal, be it a naturally exposed animal or an experimentally injected mouse or sheep or cow, etc. Very early on in the disease, depending on the type of prions, you start to see prion accumulation in lymphoid tissues after peripheral injection or oral ingestion. The disease then slowly starts to spread to the CNS. And it's not until the later stage of the disease, when you've got significant accumulation in those CNS tissues, that the animal starts to show clinical signs. Now, when might that animal be able to spread disease to other animals? That would depend on how the exposure occurred, of course. So if you were to take, for example, a sheep halfway through the incubation period, when it apparently looks clinically healthy, but if you were to grind that sheep up, make a meat and bone meal pellet, and then feed it to another animal, very likely that would transmit disease because there would be likely to be prions in that pellet. The animal doesn't have to necessarily show clinical signs to be able to transmit disease.
Which makes the epidemiology very difficult.
May, of course, be shedding feces, urine, saliva, etc. onto pasture, which might be contaminated with prions. And it's also plausible that for sheep with scrapie that disease is perhaps been transmitted to lambs around the time of birth, either through trauma, etc., during birth, or perhaps ingestion of placental or placental materials after birth, or contamination of pasture with those materials, which are then ate by other animals, etc.
So I imagine
there are multiple ways.
So I imagine this makes the epidemiology difficult, hard to trace.
Yeah, and once lymphoid tissue has actually been identified as a site of prion accumulation in variant CJD, the issue was then raised, or that actually blood transfusion might be a concern, because as you can imagine, white blood cells are present within lymphoid tissues. They might be contaminated with the prions. They'll then enter the bloodstream, and if somebody was unfortunate to receive a transmission of blood from an affected donor, they might go on to develop prion disease. And that did unfortunately turn out to be the case, despite our efforts at removing certain materials and certain individuals as blood donors. A few cases of which we most likely linked to blood transfusion have occurred. They were mostly recipients of whole blood, if I remember correctly.
Wow.
And we can trace those back because some of those individuals are known to have given blood donations, but at a time when they were clinically healthy and didn't know they had prion disease.
Of course, right. So let's
Fortunately, though, there appears to be only just a few cases of those, but it's still, of course, a big concern.
Yeah, absolutely. So let's jump forward a little bit, and I'd love to talk about what are your primary research questions today. So how sort of has the field evolved since the time that you became involved with this research?
Yeah, so the prion disease field itself, I guess, the priorities have moved as the I would say the disease itself unfolds. But my research is still trying to address that question I asked, I guess, when I first started working on this. How on earth do those prions go from the gut to the brain? And you might say, well, Neil, you've been working on this for a couple of decades now. Surely you've answered that question. But actually, the prions have been quite crafty. Firstly, they seem to hijack, I've mentioned this a couple of times now, they seem to hijack our immune systems. And the cells they're hijacking are actually cells that we knew little about. So they're not the typical T cells or B cells. They're kind of, I guess, the easier targets that an immunologist, etc., will be able to work on. They seem to hijack the cells of which at the time we knew very little. So we've had to create tools, or the field itself has had to create tools to be able to study these cells in the first place before we were able to then study their involvement in prion disease. And that's where all those collaborators I've mentioned have come into this. And it's been a really nice way to do this, because we've had this hypothesis of how the prions might be transmitted from the gut to the brain, and sequentially we've been able to know, okay, so let's look at this cell, let's look at that cell, let's look at the other cell, etc. Be able to kind of piece together a kind of chain of events. I kind of call it like a cellular relay, that the cells pass through one cell, they then pass the prions onto another cell, which then passes it onto another cell, etc., and eventually makes its way to the nervous tissues and then can spread on into the brain.
That's
and we've
amazing.
been able to pick away at that cellular relay to work out how the prions are indeed doing that. Because the reason being, if we can identify that chain of events, we can perhaps identify a way to stop it. And actually, in some of our mouse studies, we've shown actually if we remove a particular cell in that chain of events, we can indeed stop the prions getting into the body and causing disease in the first place.
Wow.
Of course, removing the cells is great in a mouse, but we need to find some way of I guess, more specifically targeting that process.
So that was going to be my next question. Are all of the cells you've found so far in this relay essential to the host, or are there any that are?
Oh, yeah.
Yeah, that's what I figured.
Yeah, exactly. And what we're actually finding is, I mean, I'm a microbiologist and I'm also immunologist as well. And if we think about how prions are orally acquired, the prions first are taken up into structures known as Peyer's patches, and these are immune kind of collections of immune cells or an immune tissue in the small intestine of the mammalian species. And these tissues are there to essentially sample the contents of the gut lumen, identify any pathogens, and make an immune response if there's a pathogen there. Now, what the prions appear to be doing is, I keep mentioning this, hijacking that essentially antigen sampling machinery. But they're doing it silently, because the host doesn't recognize the prions as foreign.
Hmm.
So they first slip through a cell known as an M cell. This is an epithelial cell which lines the gut epithelium, and it's there to sample the contents of the gut lumen and pass things through to the pairs patch so immune cells can recognize whether there's friend or foe there and mount an immune response. The prions appear to slip through these cells as well.
Hmm.
They then appear to be picked up by dendritic cells or macrophages. And they seem to be using these kind of like a Trojan horse
Wow.
because these cells move around the pairs patch and they appear to be carrying the prions then into we call the B cell follicles.
Mm-hmm.
Within the B cell follicles, prions then target what are known as follicular dendritic cells. And I also describe these as cellular spiders' webs.
Hmm.
They sit there and essentially trap and retain antigens or foreign particles. And they do so that other cells of the immune cells, particular B cells, can interrogate these kind of foreign particles and say, okay, do I need to make an antibody response to this protein or not? And by chance, these cells express really high levels of the cellular form of the prime protein. So what we have is the cells sneaking
Hmm.
through M cells, using dendritic cells as Trojan horses to reach these follicular dendritic cells, which express loads of prime protein.
Wow.
And so when the abnormal prime protein reaches the follicular dendritic cell, it then starts to amplify itself
Hmm.
on these follicular dendritic cells.
Wow.
And
And
then
it amplifies
when that happens,
the folded form.
yeah,
Wow.
exactly, yeah,
Yeah.
these seem to be the key sites of prion replication in
Wow.
lymphoid tissues. And both ourselves and other key groups around the world have been studying both of these cells and come to the similar kinds of conclusions about these cells. They appear to be kind of factories of prions in the lymphoid tissues.
Hmm.
Then once a prions reach a particular level, they can then spread. They reach a kind of a level where they can then spread into the nervous tissues because lymphoid tissues are also full of nerves. The prions don't seem to be able to make their way directly into the nerves. They seem to have to go through this kind of hop, skip, and jump through the different cells in the immune system onto the dendritic cells to reach that level, which they can then infect those nerves. And then once they do so, then start to spread quite elegantly along nerval circuits directly into the central nervous system. And so we're still
Wow.
trying to piece together that method of transmission. Look at the molecules and cells are involved, why those cells are involved, and try and target it. But of course, all that happens. All what I've described, sorry, is in the steady state
Hmm.
in what we call an SPF mouse, so a nice squeaky clean mouse in a typical mouse colony. But we know sheep in a field, humans, etc., aren't SPF.
Hmm. That's a good
We
point.
might have a bit of inflammation. We might have cold, flu, coronavirus, etc. Sheep in a field might be infected with a, a worm infection. Who
Hmm.
knows? Or
Hmm.
salmonella, etc. And all these can add stimuluses, etc., to our immune systems.
Wow. Hmm.
Likewise, aging. If we go back to, if we think about variant CJD, I mentioned it was affecting predominantly young people,
Right.
where sporadic CJD was very rare in young people. It was only a disease of old people. Now, why was variant CJD predominantly targeting young people? It still is to the case this day. It still has a meek age of incidence of about 29 years old. To
Hmm.
contrast that of sporadic CJD, which I mentioned was 60s, 70s, maybe more. Why was that disease just targeting young people?
It's a great
And so
question.
people at the time,
Hmm.
it's still an interesting question.
Yeah.
And people at the time thought, well, it's obvious. Young people eat lots of pies and burgers and that kind of stuff. And these are kind of foods which will have been packed full of BSE, and that's
Hmm.
why they're
Hmm.
developing food. They develop variant CUD. When you think about it, actually, well, I know lots of old people also eat pies and burgers
Right.
and sausages, etc. Yeah. So
Hmm.
why aren't these guys coming down? And actually, when epidemiologists looked at this, it's not quite as straightforward as that. There's definitely a dietary link, but
Hmm.
it's not simply young people eat more pies than old people, etc. So we thought, well, what about the immune system? We know as we get older, our immune system starts to become less effective. We call that immunosenescence. We know we don't have to look to the coronavirus pandemic for that, whereas older people were much more susceptible to the adverse and serious illness than young people, weren't they? And were given priority for vaccination, etc.
Mm-hmm.
We wondered, well, maybe there's a link between the impacts of age on our immune system and susceptibility to prion disease. Because I mentioned the prions have to use particular cells to enter the body. And so
Mm-hmm.
we looked at the cells that the prions hijack to get into the body and realized actually, well, if we look at old animals, there are much fewer of those cells, or those cells don't work as effectively in an older animal compared to a younger animal. It may be actually that the effects of age on the immune system are impeding the ability of the prions to infect the body, and that might link to why these diseases are more prevalent in younger people than they were in older people. So we're also trying to look
Mm-hmm.
at how other factors can influence susceptibility, because one of the early questions we mentioned was trying to gauge how many people develop variant CGD. Would it be tens of people, hundreds of people, or millions of
Mm-hmm.
people?
Mm-hmm.
And I would say, fortunately, there's been around about 180 people, at least in the UK, have succumbed to variant CGD. Still a large amount, but fortunately it was restricted to that because it could have been a lot more.
Yeah.
But why so few? Perhaps maybe there were additional factors which may have made some people more susceptible than others. So we've been looking at
Right.
the impact of stimulating the immune system.
Mm.
Does a co-infection, etc., make it more likely that a host becomes infected or not, of course? So there are many different kinds of questions now which we're trying to answer, but
It's
around
so
this central theme of how the prions spread to the brain.
Yeah, it's so fascinating. I could talk to you about this all day. I really have enjoyed this conversation. So thank you for sharing both the history and also your experience in this field. I'm curious, kind of, we touched a little bit on the fact that it sounds like there is not a treatment currently, and that this is ultimately fatal for patients. Um, but I guess, can we talk a little bit about that? What is the method of care? Is there any prevention that we know of now? What can we do essentially best to prevent and stop this?
Yeah, I mean that's the frustrating thing about this
Mm-hmm.
is that we still don't have a cure for this. That's not for lack of trying, and there are
Right.
some really novel methods being looked into this. I mean, if we look across, of course, to the Alzheimer's field, and some would argue that prion disease is reasonably similar to Alzheimer's disease in some of the
Mm-hmm.
neuropathological signs. So attempts are being made to look at what would antibodies work for this type of disease. Say, for example, if we give a prion-infected person an antibody against the prion disease or the abnormal prion, sorry, will that enable the immune system to clear up those prions and at least help stop the progression of the disease any further? And there are some suggestions that this might work, but of course we have to use these with caution because, as I mentioned, every cell in our body expresses a prion protein. So we have to be very careful that the antibodies we inject don't also target our own body cells and
Right.
help to destroy those. And the last thing we want to do is inject an antibody which is going to target normal, healthy neurons and kill them.
Right. Oh my
So
gosh.
these are being trialed both experimentally, and there have been some limited clinical trials done on this,
Mm-hmm.
not least to look at just the safety efficacy of those. But we're not at the stage yet that we can do that. Others are looking for small molecule, I guess, inhibitors. Can we identify some little small molecule we can give somebody that might actually prevent that conversion happening? So it might bind to a binding site, perhaps between the normal prion and the abnormal prion, and perhaps stop that
Mm-hmm.
happening, so we can stop the disease. But as you're probably learning, though, a lot of these treatments that I'm talking about are stopping the disease progressing further,
Right.
because what we don't have yet, likewise, is a reliable diagnostic test. So we need
Mm-hmm.
a diagnostic test which could work really early into disease, that has very little, I guess, negative detection rate, etc. A very highly selective one that we could then target these therapies too, because the sooner we can stop the disease, the sooner we can prevent the severe neurological damage happening in these individuals. Of course, it'd be nice if we could have a vaccine against prions, but again, there are the similar concerns that you need to make sure that these antibodies don't target the normal healthy cells in the body. So for those individuals which unfortunately do go on to develop prion disease, it does come down to, I guess, palliative help and making sure those individuals are most comfortable for the remainder of those disease. And that's the really unfortunate thing. Of course, if we do have a diagnostic test and we don't have a treatment, of course, so we would identify people that do have disease. But then there's the ethical concern. Okay, well, how do we manage those people? And we say, oh, unfortunately, you have prion disease, but there's nothing we can do about it at this stage.
Right.
So
Right.
there is this real area of active research is trying to look at a therapy for these diseases, but we certainly aren't at the stage where we can roll one out safely too. That's not, like I say, it's not to say that things haven't been done.
So when it comes
it comes back to reducing risk of transmission. And I mentioned, for example, when there was a relative risk of blood transfusion was identified, steps were taken to eliminate aspects that were most likely to be infectious, etc.
And it sounds like changes also agriculturally, that there have been changes as well to change the feeding approach.
Yeah, changing farming practices, removing things from the food chain, etc.
So when it comes to development of a targeted diagnostic approach, obviously we can't do nucleic acid testing. What are some of the methods that are being investigated there? Is it an antibody antigen approach or
for a diagnostic test? So people have tried ELISA style based tests, but of course what we want to look at is we want to be able to detect prions in an easily accessible sample. So people are unlikely to say, can I drill a hole into your head so I can get a little tiny piece of brain, and then run a Western blot, etc. People aren't going to really come forward for those kind of tests. So we want something which we can identify perhaps in a small sample of blood, maybe in urine, or maybe a bit of sputum, etc. But we do so, we have to bear in mind that there'll be very, very, very low levels of prions there.
Okay.
Talking incredibly small amounts of prions compared to what you might find in a lymphoid tissue sample or in a brain sample. So we need a really, really sensitive test here. And so tests are in process, and they're mostly tended to be based now on those, I call it the PCR for prions kind of test, where you have serial rounds of adding your sample to a preparation of abnormal prion proteins. You hope that if there are prions in your test, it'll convert some of those normal prion proteins into abnormal prion proteins, and by doing multiple rounds of this assay, you can really amplify a tiny amount to a quite large amount that you can then detect on using a Western blot or some similar kind of assay. We have to be very careful with this though, because these tests experimentally are very, very, very sensitive. And we need to be careful that we don't generate false positives, but because of course there could be quite serious implications of generating these false positives. But there is some good progress being made in that, but we need to be very careful about that. We also need a quick test as well. We need a nice rapid test as well. So if we have a load of blood samples, we can't wait several weeks until we get a positive test, can we? We need to know that this is a reliable test and get our results quickly.
Right.
We also need to know, of course, that this abnormal folded protein that we are detecting is actually prions. and not just some abnormally folded protein that happened to be in the body. So again, we
Right.
need to do lots of validation on these kind of things. We can rule out all these different caveats. We have a nice, robust, reliable test that's
Makes
highly
sense.
sensitive.
So, Neil, what is next for your research? What do you see for the next 5 to 10 years?
That's a really good question, and I rarely know what I'm doing next year, let alone in the next 5 or 10 years,
Yeah.
because there's always something new
Hmm.
in science. For my next 5 to 10 years, I think it would be really nice to have drilled down to that cellular relay,
Hmm.
have identified the key cells, have identified some aspect that we could then say, okay, if we target this point by this mechanism, we might be able to prevent animals becoming infected with disease. And here is an element where, like genome editing, could
Mm-hmm.
be a real game changer here, in that we may be able to identify a receptor which the prions are exploiting on one of these different types of cells. And by just making a slight modification in the binding site of that receptor that the prions are using, we might be able to create a breed of cows that no longer can be infected with BSE, for example. And that would be
Mm-hmm.
a really nice way of being able to do that. It may similarly be able to target the prion protein itself, and to be able to specifically target a region of the prion protein and use through gene editing techniques, which are advancing at rapid pace now. And my colleagues at the Roslin Institute in Edinburgh here are experts in this, so we could quite easily be able to use this technology to certainly improve food security.
Mm-hmm. And what is it about your work and your research that you find most rewarding?
I always find the surprises the most rewarding things when you say, "Oh, we
Mm,
didn't expect that."
I
Often we
love
make
that.
hypotheses that if this cell is really going to be really important, we do the experiments, and yet it turns out that this cell, we find the circumstances we expected. But oftentimes those sometimes frustrating results, but they can be the most exciting ones when the student comes back to you with data. Well, I did the experiment, Neil. These are your data. Are you sure? You sure that's
Yeah.
go away and do it again? They come
Yeah.
back and they get the same things. And well, okay, well, we weren't expecting that, but oftentimes that uncovers something really, really interesting that we hadn't thought about, and often opens up some really exciting areas. So
Yeah.
they're the kind of things that keep me going. And often that usually means, okay, well, we need to speak to somebody about this because I know nothing about that particular cell
Hmm.
or that particular pathway, etc. So I need to speak to another collaborator now to find out what we're going to do about that. And then that will often set up some new discussions and some new exciting challenges leading to hopefully nice papers, etc.
Oh, that's so neat. And what advice would you give to early career microbiologists?
Yeah, that's a really great question, and there are multiple ways, things I would say to do that. Uh, Firstly, I think it comes back to recognizing the opportunities you get.
Mm-hmm.
People always say, well, so and so was lucky, and so and so probably was lucky, but they knew when that luck arose. So always make use of those opportunities that come across. Don't
Yeah.
sit on them, etc. Like hopefully I've explained during this
Mm-hmm.
discussion. I didn't start on prions because I knew it was going to be a disease which in a year or so of starting was going to be have such high
Yeah.
public health and food security relevance, but it did, and I was able to use that opportunity for in many ways. So make use of those opportunities. Don't be shy to ask for help. Oftentimes, early careers researchers, especially PhD students, unfortunately, seem to think when they join the lab that they're expected to be the experts. It's like, no, you're
Yeah.
here to learn and train, etc. Yeah. So
Mm-hmm.
if you don't know something, ask. There's bound to be somebody nearby that has expertise, etc., which is able to help you.
Mm-hmm.
Should you need it, ask for help through collaborations, etc. I have throughout my career and always been delighted in the ways that people have responded to me for those requests. And I would say to those early careers, when you become senior researchers, think about the help you got along the way as well. So when somebody comes to you for asking for, oh, can I collaborate with you? Instead of saying, oh, I'm not interested, etc. Yeah, yeah.
Yeah.
Take the opportunity to at least speak to those people, and if you can help them, because you'll both benefit uh, from that.
Neil, thank you so much for speaking with me today and for taking the time to discuss the complex host microbe interactions of prions and prion disease. It's been such an informative and enjoyable conversation. I've really had a great time speaking with you.
Thanks, Ashley. It's been a pleasure speaking to you as well.
That's it for today's show. If you enjoyed this episode, please share it with someone who might also enjoy it. I'd like to thank Ray Ortega for our podcast production. I'm Ashley Hagen, and I'll be with you next time with a new microbiologist for you to meet. All right, if we could just hold off.